Hello to all of you. Now we're going to discuss chapter number 12. And in chapter number 12, we are going to discuss about the process of translation. So this is the second step in the gene expression. So first of all, in this chapter, we are going to talk about the different components which are required in this process, which involves transfer RNA, ribosomes. So first of all, we will talk about what they are, how they are, what is the structure. Then we will talk about the process of translation. So let me go into the slideshow mode. Okay, so so we have already discussed that how mRNA is formed, and then now this mRNA will be going from the nucleus to the cytoplasm, and there it is going to go and look for the ribosomes, and on the ribosomes, so this mRNA is going to go and bind on the ribosomes, and these ribosomes actually they move on the mRNA, and then a protein, a polypeptide chain will be formed on the ribosomes, and this process involves another kind of RNA which is called transfer RNA. And in this, so basically the process of translation also has three stages: initiation, elongation, and termination. And in every step, there are some proteins involved, some factors involved in every step. So first of all, the transfer RNAs, which I just said, they are transfer RNA is that RNA which is responsible for transferring the amino acid. So transfer RNA is that RNA which is responsible for transferring the amino acids, and these amino acids then they form peptide bonds and they form the polypeptide chain, and these polypeptide chains then they undergo further post translational changes, and this polypeptide chain folds and it forms a three-dimensional structure of the protein. So these transfer RNAs, first of all, they are activated, and they are activated by the involving the enzymes called amino acyl tRNA synthetases. So these transfer RNAs, they have a code on them which we call it a anticodon, which is basically recognized by a codon, and codon is on the mRNA. I'm going to discuss about these things. So basically, this transfer RNA, when it is without amino acid, it is called that transfer RNA is uncharged. When the transfer RNA gets the amino acid, when the amino acid binds to the transfer RNA, then that transfer RNA becomes charged, and that charging of the transfer RNA takes place with the help of this enzyme called amino acyl tRNA synthetase. And as I said, the process of translation involves three steps: chain initiation, elongation, and termination. So initiation, elongation, and termination. So first of all, let's talk. So before we actually start talking about the process of translation in three steps, which involves initiation, elongation, and termination, I'm going to talk about some terminology, because only then you will be able to understand. So first of all, what is the triplet code? So triplet code or codon, this is what is a codon. So wherever the word comes codon, remember that codon word refers to mRNA, and the word anticodon refers to word transfer RNA. So what is a codon? It's a sequence of three bases. This is a sequence of three bases which is there on the mRNA. Okay, and from where this information, or from where this codon is coming on mRNA, it is coming basically from the DNA. Okay, so you know that the DNA gets converted into mRNA, then mRNA gets converted into proteins. So the information basically is coming from DNA, and the three bases, three ribonucleotides on mRNA, they form one codon. And every codon, which is a set of three bases, we also call it a triplet code, it codes for one amino acid. And that one amino acid is brought by a transfer RNA. So transfer RNA has an amino acid on one side, and it has an anticodon on the other side. I'm going to show you that. And this anticodon, which is there on the transfer RNA, this anticodon which is there on the transfer RNA, it is recognized, it recognizes a codon on the mRNA. And on the basis of the codon on the mRNA, the anticodon Anticodon is complementary to the codon. On the basis of that, the transfer RNA has the specific amino acid on it. So what is the triplet code? Sequence of 3 bases, which we call codon, is needed to specify one amino acid. Okay. These bases, they are not non-overlapping. So basically no bases are shared between consecutive codons. So three ribonucleotides form one codon, and every codon codes for one amino acid. So it's a commaless. The codon is commaless, which means there are no intervening bases between the codons exist. So if we have a sequence of mRNA, so first three ribonucleotides makes one codon, then the next three makes another codon, then the next three makes another codon. So this is the way the codons are there, and there are around 64. There are around 64 triplets. So there are actually 60. There are around 64 codons, out of which 3 codons are called stop codons. They are called UAA, UAG, and UGA. These are called termination, or they are called the stop codons, and they are the ones which are so these are the codons which are called stop codons, and they are the ones which signal the translation machinery to stop the process of translation. And these 61 codons, they code for amino acids. So this is a codon table, or this is also called genetic code. So these are all ribonucleotides, or these are codons which are present on mRNA. Okay, so you can see here, like wherever in the mRNA there is a UUU, it is going to code for this amino acid called phenylalanine. If this is UUC, it's again going to code for phenylalanine. UUA, leucine. UUG, leucine. CUU, leucine. So this is the way, this is called the genetic code. Okay. So, and these are, there are 3 codons, UAG, UGA, and UA, these are stop codons, and this is AUG. AUG is a start codon. Okay, AUG is a start codon. So on the basis of that, on the basis of this codon table or genetic code, the proteins are formed in the process of translation. There are 4 amino acids, and there are around 45 transfer RNAs, which means that there are so there are 45 transfer RNAs and 61 codons, which means that some of the transfer RNAs can actually code for more than one codon. So, and that is why we call genetic code is degenerate. So degenerate code, more than one triplet can encode the same amino acid. So as I said, one transfer RNA can code for more than one can code for more than one codon, which means that every transfer RNA is bringing one specific amino acid. So every transfer RNA is bringing one specific amino acid, which means that one transfer RNA can actually recognize more than one codon. Okay, so if there are 61 codons and there are 45 transfer RNA, which means that one transfer RNA can recognize more than one codon, and which means that some of the codons can code for same amino acid, like UUU can code for phenylalanine, UUC can also code for phenylalanine. Then there are 6 codons. Here are 6 codons which are 6 codons which code for the amino acid called leucine. So UUA, UUG. So these are all codons which code for the same amino acid. Okay. So that is why genetic code is called, it is degenerate. Universality of the codon. Universality of the codon means this codon table is actually followed by all the organisms in this whole universe. There are only few exceptions. Okay, so that is why genetic code is universal in nature. So codon can form base pairs with complementary anticodon of a transfer RNA. So let me show you actually the structure of transfer RNA. So this is the structure of transfer RNA. Okay, So to make it simple, there is 3 prime end on the transfer RNA where the amino acid is bound, and then this is the part of the transfer RNA where there is a anticodon, and this anticodon is actually going to recognize the codon or codon on mRNA. So going back, so anticodon is on transfer RNA and codon is there on the mRNA. So some transfer RNAs bound to one codon exclusively, but many transfer RNAs can recognize more than one codon. Now, as I said, there are some codons which can code for same amino acid. Now, if you look here, these are the codons. So these are the codons. These are different codons, but they code for the same amino acid. So there are some transfer RNAs which can recognize only one codon, but there are some transfer RNAs which can recognize more than one codon. Now, how one transfer RNA which has one anticodon, how a transfer RNA with one anticodon can recognize more than one codon? The answer is Wubel. Okay, this is Wubel word is coming from the name of a scientist who found that the sum of the transfer RNAs on the 5 prime side, so this is the anticodon in a transfer RNA, on the 5 prime side, the nucleotide they have, this nucleotide is actually modified. This nucleotide is modified, and as a result of that, they can actually recognize different nucleotides in the mRNA. So you can see here some of the transfer RNAs in their anticodon on the 5 prime side, they have the nucleotide is I. This is basically a modified nucleotide. It can recognize A, C or U. Okay, so that is why if it is I here, it can recognize A, it can recognize C, or it can recognize U. Okay, and then like some of the transfer RNAs, they have the last nucleotide is G. It can either recognize C or it can recognize U. If it is U, it can either recognize A or G. So this is the reason. This is called Wubel effect or Wubel scheme. So on the basis of this, one transfer RNA which has one anticodon can recognize multiple codons, and in this table you can see that some of the amino acids, they are coded by multiple codons. Okay, so transfer RNA is bringing the same amino acid. Okay, it's bringing the same amino acid, but it can recognize more than one codon, and that is why there are multiple codons for amino acids. I hope it makes sense. So that is why we call it genetic code is degenerate. We call it universal because almost all organisms in this whole universe, they follow the same rules. Okay, I covered this. So I have explained this. This is all coming here. Now now we next need to know the structure of ribosomes. So ribosomes, they are made of two subunits. So they are made of two components. They are made of rRNA, which is a ribosomal RNA, and they are made of proteins. So ribosomes are complex structures which are made of proteins, and they are made of ribosomal RNA. And they have two subunits. They have one big subunit and then they have one small subunit. And in the prokaryotes and the eukaryotes, the ribosomes are different. And the ribosomes, they have like there are two subunits, one is small, another is big. The bigger subunit has three sites: P site, A site and the E site. Okay, so we will be talking about these sites. So E site is called the exit site, from where the transfer RNA which has delivered its amino acid, the transfer RNA exits from here. A site is a site on the ribosome where the new transfer RNA enters, and P site is that site on the ribosome, on the bigger subunit of the ribosome, where the growing, where the transfer RNA is there, on which the polypeptide chain is growing. So this is the structure of ribosome. This is the smaller subunit of the ribosome. This is the bigger subunit of the ribosome, and this is the whole complex of the ribosomes. And these are the three sites on the bigger subunit of ribosome: A site, P site, and the E site. This is the exit site. This is the site from where the polypeptide chain grows, and this is the A site where the new transfer RNA enters, which brings the new amino acid to the chain. So this is the transfer RNAs. You can see this is the transfer RNA which enters on the A site. This is the transfer RNA where the chain of the amino acids, the polypeptide chain is growing, and this is the E site from where the transfer RNA which has given its amino acid, where E site is the site from where the uncharged transfer RNA which has delivered its amino acid to this transfer RNA, it goes out. So now I think we are ready to discuss the process of translation. So there are three stages in the process of translation: initiation, elongation, and termination. So before we can understand these three stages of the translation, we need to know the structure of transfer RNA, we need to know the structure of ribosomes, we need to know the mRNA which is formed by transcription. It has codons. Transfer RNA has an anticodon, and codon and anticodon, they can complementary to each other, and on the other side of the transfer RNA is a amino acid. And transfer RNAs, when they are without amino acids, they are called uncharged, and when they get the amino acid attached to them, they are called the charged amino acids. So now let's talk about how the process of translation starts. So we know that when the mRNA is formed, it's modified, it reaches the cytoplasm. First of all, it goes and binds to the smaller subunit of the ribosome. Something very important to know about this process in the initiation process is the two subunits of the ribosome, the two subunits of the ribosomes, they are not together like that, they're not associated like that all the time. They are actually separate, they're disassociated. So mRNA goes and binds on the smaller subunit of the ribosome. So then this ribosome, the smaller subunit of the ribosome, it actually walks on the mRNA, okay? And when it actually walks on the mRNA, it is looking for the start codon. The start codon is AUG. In every mRNA, the start codon is AUG. So once this ribosome finds the AUG, the bigger subunit of ribosome is also gets associated to the smaller subunit, and this kind of a complex is formed. Once the smaller subunit of ribosome is going to go and walk on the mRNA, and it is going to it will be walking on the mRNA, and it is looking for AUG. Once it finds the AUG, there is a recursion of this transfer RNA which is having methionine on this side, okay? And also there is an association of this larger subunit of the ribosome, and this is the translation initiation complex which is formed. Important thing to know here is on the mRNA, the start codon is AUG, this is the start codon, and on the transfer RNA, the anticodon is UAC. So this is translation initiation complex which is formed, and this transfer RNA which is having methionine on this side, it is placed on the P site. It is placed on the P site. So this is the A site, this is the P site, and this is the E site. And after initiation, so this is the complex which is formed, okay? Now this site is free. Now this ribosome as a full complex, this ribosome as a full complex actually walks. This ribosome as a full complex, it actually walks on the mRNA, and then the next codon is going to be read. And when this next codon is going to be read, there will be recursion of whatever the codon is here. On the basis of that codon, a new tRNA will be coming here, and it will be sitting here, okay? And that transfer RNA will be having a complementary anticodon. So here is a codon, that transfer RNA will be having a anticodon, okay, and on the basis of this codon and the anticodon it is going to bring the specific amino acid. So let me show you here, and this is all part of the elongation process. So here you can see this is the transfer RNA. So here they have shown you here a transfer RNA with few amino acids. So let's imagine there's only one amino acid here, which is methionine, so which is present on the P site. A site is free. Now whatever the codon is here, the next transfer RNA is going to bring the next amino acid, and it will be having a complementary anticodon. It's going to bind the it is going to recognize the codon here, and it is going to bring the next it is going to bring this transfer RNA here, which is having a specific amino acid, which is being on the basis of the table. You can see whatever the codon is here. Suppose I'm just making up, if the codon is G, suppose it is GGG, the anticodon here will be CCC. And suppose GGG on the basis of the table, I'm just making up, if GGG codes for serine, the amino acid is going to be serine here. And this transfer RNA is going to come and bind here on the A site. So there is one transfer RNA here, which is having methionine. There is a next transfer RNA here, which is having, suppose this is serine. Okay. So after that, what will happen will be this transfer RNA, which is having a methionine here or a growing chain here, it is going to break its bond. This transfer RNA is going to break its bond with these amino acids, and it is, so this transfer RNA, when it breaks its bond with these, when this transfer RNA breaks its bonds with these amino acids, it is going to become uncharged. Okay, when it becomes uncharged, it moves from this side to this side. This is called translocation. Okay, this transfer RNA moves from this side to the exit side, and this transfer RNA and this amino acid, which is here, it will form a covalent bond with this amino acid. Okay, so whatever the amino acid is here, it is going to make a bond with this amino acid, and this amino acid is actually bound to this transfer RNA. So when this uncharged transfer RNA moves from here to here, okay, it moves from here to here. Sorry. So this transfer RNA moves from this transfer RNA, which is having these amino acids here, it will move from A site to P site. This is called translocation. The transfer RNA, uncharged transfer RNA from P site, it moves to E site, and the transfer RNA, which is having the growing chain of amino acids, move from A site to P site. Okay, this is all part of translocation. This transfer RNA from the E site, it is going to go out of this ribosome. Okay, it is going to move out from here. It is called, that's why it's called exit site. And this transfer RNA is having the growing chain of amino acids here, and that is why this site is called P site, because the growing polypeptide chain is here. And A site is always free. A site is always free because A site is always free, so that the new transfer RNAs can keep coming here, and they keep bringing new amino acids here. Okay, so this is all elongation, and this will keep on going till the ribosome finds the stop codon. So this is all part of elongation, and this process will stop when the translation machinery finds a stop codon. Now the stop codon is UAG, UAA and UGA. Okay, so once the translation machinery finds a stop codon, at that point on the A site is a binding of a protein called release factor. And when the release factor comes and binds on the A site, as a result of that, what happens is the transfer RNA, which is sitting here on the P site, it breaks its bond with this peptide. This peptide is released, and this transfer RNA, this peptide is released, and this transfer RNA moves from P site to E site. It moves from P site to E site. That's the exit site. Okay, so once the translation machinery finds the stop codons, it can be either of these three. The release here, this transfer RNA breaks its bond with the peptide, and this transfer RNA moves from P site to E side it goes out. So peptide is released, transfer RNA is out, and these two subunits of the ribosome, they again get disassociated. And this way the process of translation stops. So by this polypeptide chain has formed. And you know that we have studied in the structure of proteins, the chain of polypeptides, this amino acids linked to each other, they form a polypeptide chain. So after that, this polypeptide chain needs further processing, it needs further modification, only then the final three-dimensional structure of protein is formed. And these kind of changes, they are called post-translational changes, which takes place after this peptide is formed. And also in prokaryotes as well as in the eukaryotes, to the mRNA, multiple ribosomes can bind, and and they can start the process of translation. So you can see here, this is mRNA, there's one ribosome binding here, there's second ribosome binding here, there's third ribosome binding here, there's a fourth ribosome binding here, and the ribosomes, they actually move on the mRNA and they keep making polypeptide chains. So you can see here, these ribosomes, they are moving on the polypeptide chain, these ribosomes are moving on the polypeptide chain, and they are forming the peptide. So this ribosome has reached here, the peptide is formed. So this ribosome has reached here, this much peptide is formed. This ribosome has reached here, this much peptide is formed. So this is called polyribosome. And what's the significance of polyribosome? The significance of this is, by this, the efficiency of translation can be increased and more protein can be made in a short time. So that's all what we need to know from this chapter. If you have any question, please let me know.